Molecular Cytogenetics, Module 1, Lecture 7 in the Genetics series. Molecular cytogenetics, routine cytogenetic analysis can detect approximately 5 to 10 megabases, a deletion or duplication of genetic material. High-resolution analysis can detect deletions in about the 2 to 3 megabase range. Deletions smaller than 2 megabases are extremely difficult to detect using cytogenetic techniques. Fluorescent in situ hybridization, or FISH, was a method that we've had for approximately 20 years, and it's been helpful in trying to bridge the gap of further detection. Basically, a fluorescent probe is used to label a single-stranded DNA. It allows for better resolution of chromosome deletions and translocations. Unlike southern analysis, DNA is not extracted and run out of gel, but it's studied right in either a metaphase spread or even the nucleus of the cell. In the procedure of FISH, or fluorescent in situ hybridization, the chromosome is denatured with heat or formamide to denature the double-stranded helix and make it single-stranded. A target DNA is now available for binding to the complementary sequence. A probe is tagged with a hapten such as biotin or digoxin. Following the hybridization, the hapten is labeled with an antibody or fluorescent dye. So here we have a diagram of our probe that we label and we attach to the area of interest. If it attaches, it's there, and it emits its fluorescence. If it's not there, it doesn't have anything to attach to. So if there's a deletion in the region, this specific probe does not attach. Again, this is useful in both interphase nucleus. Here is some uncultured amniocyte cells where there's a concern that the baby may have Down syndrome. A specific probe for repetitive DNA on chromosome 21 is used. And here we have 3 copies of chromosome 21, and here we have a metaphase spread. This can also be done for microdeletions, or very small pieces on a chromosome. The yellow arrow represents the critical region that we're interested in, and because these blue blobby things you could not tell between chromosome 15, 16, or 21, this tells us that in this case it's chromosome 15. Here we have 2 copies of chromosome 15. Here's the critical region, but here it's missing. We can look at 200 cells, so it's not just a single cell, but we look at hundreds of cells, and when there's no attachment, that means that there's a deletion in that region. These are, as stated, deleted regions, and they're commonly used for specific disorders. The disorders that can be used can be diagnosed using this technology. Fluorescent in situ hybridization includes small deletions on chromosome 15, including both Prader, Willi, and Angelman syndrome. The same probe is actually used for both, even though they're separate disorders. 22q11 or velocardiofacial, Williams syndrome 7q11.23, Smith and McGinnis 17p11.2, and Miller-Duker, which is 17p but 13. These are actually different probes, whereas up here these are the same probe. And while this is a very helpful method, there's a limited number of disorders that it can be useful for. In the case of Prader-Willi syndrome, 70% of patients with Prader-Willi will have a deletion in the 15q11 region. This is usually only identified using FISH, so if we looked at cytogenetic analysis, we would not see this very, very small piece missing. 30% are caused by both copies of chromosome 15 coming from the same parent, which is known as uniparental disomy, and in that case, the FISH would actually be normal. Microarray is a new technology that's been available for the past 6 to 7 years and has gained promise and is now really the method of first line in patients with multiple anomalies, cognitive delays that do not fit into a specific disorder, for instance, Down syndrome. With comparative genomic hybridization, we have a slide, but really the slide is more of a computer chip that each one of these wells represents a certain area of a certain chromosome. We have our patient DNA and our control DNA, and we add it, and what we expect is equal hybridization to each of these levels, and that means that there's no deletion or duplication. If there's more patient DNA, that indicates a duplication at that specific region, and if there's a loss of patient DNA, that means a deletion. So the DNA from the patient and the control are looked at. Both additional and missing material can be detected throughout the genome. So with FISH, we need to know what we're looking at. With microarray, we can analyze the entire genome using this technology. When a baby has multiple congenital anomalies and does not fit into an identifiable syndrome, this is actually the best method to start with. Over 100 genes need to be deleted or duplicated prior to seeing it under routine cytogenetic analysis. Microarray allows detections of either duplication or deletions at 50 to 100 megabases, and that again could be within the size of the gene. These deletions and duplications are then confirmed by FISH analysis. So here we have another diagram. Here are our patient DNA labeled with a green fluorescent probe and our controlled DNA labeled with a red fluorescent probe. Here we have our microarray slide. Again, this is more of a computer chip with each one of these wells representing a certain area of a certain chromosome. If we think of a chromosome like an encyclopedia, the genes are specific regions. We're not looking for gene sequence, but whether there's enough areas in that region to attach. So we have our patient DNA and our control DNA. We add it onto this slide. This is what we would normally get, is equal hybridization at all chromosome regions of all the different chromosomes. When we get this, we can blow that up and find out exactly where and what, and then we can confirm it with FISH. In this case, we see a little bump on 17p11.2, and then we can use FISH to prove that that region is actually deleted. So here's another diagram. Our control DNA, our patient DNA. Each one of these wells is individually analyzed through mass spec. Equal hybridization is yellow. If we see red, that means there's more control than patient, and that indicates a deletion. And if we have gain, that means more patient than control, and that's a duplication. So with FISH, we need to know what we're fishing for. We need to know what we're looking for, because we have a specific clone of DNA that we know that we're worried about, and we attach it to the metaphase chromosomes. And if it attaches, it's there, and if it doesn't, it's not. But microarray actually allows us to look at the entire genome with this single test. So here we have a little video. Targeted coverage of Prader Willi Angelman region. Here we have Prader Willi Angelman is on the long arm of chromosome 15. 70% of patients have a deletion. And while we can use FISH, if we're thinking it might be Prader Willi or Angelman, we're not sure, we can use this technology. This allows the entire genome. New words that are coming out with this whole technology include something called copy number variant, or CNV. This can indicate a loss or deletion, a gain or duplication, or my all-time favorite, bane of my existence, is variant of unknown clinical significance. There's a small deletion or duplication, but we're not really sure what it means. As we've been doing this more and more, we find more things that are called benign polymorphisms, changes that are very common in the population, therefore are not of clinical significance. So we're hoping more of this will go down to over here. Now the nomenclature for this new technology is also very important. You've learned since probably high school 46XX equals a female and 46XY is a male. With microarray, however, it'll say array 1 times 22, so 2 copies of all the autosomes and 2 copies of the X. That's a normal female. Array 1 times 22 times 2, 2 copies of all the autosomes. XY times 1, 1 copy of X, 1 copy of Y. That is a normal male. In abnormal results, only the array that's abnormal is indicated in the nomenclature, so array 2Q22.3. Then we have a series of 6 numbers. This is kind of like a zip code that tells us exactly, we know it's on chromosome 2, and this tells us exactly where. If it says times 1, it's a deletion. If it says times 3, it's a duplication. And this is, we can put this into computer databases and determine which genes are extra or missing using this. Another thing that CGH arrays will pick up is something called consanguinity. And this will indicate it as HMZ. It doesn't mean anything extra or missing, it just means it's the same. When there's consanguinity, this is often expected, but when we see small areas of HMZ, we can look at which genes are involved in them. If they're the same and both are not working, that could explain the phenotype. So routine cytogenetic analysis can identify a cause in approximately 5% of individuals with congenital anomalies not fitting into an identifiable disorder. Microarray adds an additional 9 to 15%. This microarray offers a 15 to 20% yield over routine cytogenetic analysis when looking at an individual with unexplained delays or anomalies. 20% of patients with isolated autism also are found to have a significant change on microarray. Limitations of array: it will not pick up a balanced chromosome rearrangement because the DNA there's the right amount, it's just not in the right place. The other problem is 80% of our genome is non-coding, so small additions and deletions may not be of clinical significance. Here's a question. A 5-year-old male has been diagnosed with autism and attempts to determine a possible cause. The following studies are recommended: routine chromosome analysis with FISH for 7q, 15q, and 22. Array CGH or genetic testing is not recommended.